Hi folks, and today we'll be discussing chapter 18, which is the concepts of lipids. So last chapter we spoke about carbohydrates, which was our first macromolecule. This is the second macromolecule that we'll be discussing, lipids. So just like we did with the previous chapter, we'll go through kind of understanding what they are, how we draw them, as well as things like characteristics of them, key reactions, as well as some categories that fall. Different types of steroids and things would fall in this as well. So we're going to have two kind of flavors of our lipids. We'll have saponifiable and non-saponifiable lipids, and under that we could have simple or complex. So here, as we've had before, is kind of a nice roadmap of where we're going. So the two basic categories of lipids fall under saponifiable or non-saponifiable. Non-saponifiable will be more what we think of as biological signalings in our bodies, so things like steroids and prostaglandins. Saponifiable lipids will be more of what we think of in terms of fats that we see in our diet and things like that. So simple and complex, we have lactose, triglycerides, phosphoglycerides, and single lipids. So what is the function of a lipid? We've all probably experienced fat of some sort or another. Oops, sorry about that. Fats of one sort or another in our day-to-day lives, especially if you've ever cooked meat. You know that fat tends to repel water. This is also something we witness if you look at like the skin of an apple. Granted, the ones that you find at the grocery store have an extra waxy layer on them, but natural fruits and vegetables often do have a waxy kind of protected seal from them. You'll also see this on the leaves of trees and plants. They're typically very energetically rich for their relatively low densities. It's a form of storage for energy in plants and animals. They also have structural components. So when we're thinking of a lipid membrane, bilipid membrane, bilayer lipid membrane in a cell. So the building block of all lipids is going to be fatty acids. Now, hopefully this looks somewhat familiar from our Unit 2 days. We were talking about carboxylic acids. Carboxylic acids are essentially fatty acids, just where the chain on the carbon chain is much, much longer. Typically, we'll see somewhere between 12 and 25 long chain long carbon chain long tails. So the way we describe a fatty acid is we have a polar hydrophilic head. So this is the part of the compound that is water soluble, and then a nonpolar hydrophobic tail. So here's just kind of a cartoon representation of that. So we can have something occur which we would call a micelle, and these would be spherical clusters of these carboxylic acids. In the picture here, this looks like a disc. Try and think of this more as a sphere, right? So this is like a 3D, this is just like a crosscut of one of these. But think of it as like a 3D sphere, where in 3D space, all of the hydrophilic tails, or excuse me, hydrophilic heads are outward facing, and all the hydrophobic tails are inward facing. So the inward facing tails are all completely insulated and isolated from the surrounding, typically water rich environment. If we're thinking of things like our plasma and blood and humans in general are mostly water. So these are a way to be able to transport insoluble lipids through something, through a media that is typically water, where those wouldn't mix very well. So as we mentioned before, the typical fatty acid will be a carboxylic acid that is straight chained, no branching, somewhere between 10 and 20 carbons long. Typically, they have an even number. There are a couple oddballs that have odd numbers. They can be saturated or they could be unsaturated. If they are unsaturated, it's typically a cis type double bond. So again, think back to our unit 2 where we talked about the difference between cis and trans. We will talk about the concept of trans fats in a little bit. You probably have heard of that term before, but the trans fats that we're talking about would be the opposite of a cis fat. So an unsaturated fat contains double bonds, at least one, typically more, and that typically in nature they are cis in their formation, but sometimes we can also experience trans configuration as well. So here's just a list of some important fatty acids that we see in our day-to-day lives. For example, stearic acid would be the fat that you see on things like animal meat. Oleic acid, again, would be another one of those. We also have things like the pentanoic anoic acids and the hexanoic acids, and these are fish oils and things like that. You do not need to memorize any of these. I would recommend this be a very good resource page to have available for yourself during assessments. So when we have unsaturated fats, these are the fats that contain double bonds. We mentioned earlier that most of them are cis in their configuration. If you think about what that physically would look like, a cis double bond would cause what we consider like a kink or a bend in that long fatty acid chain. And if our tail is all bendy and kinky, it can't lay flat. And if it can't lay flat, that means that our fatty acids would not be able to stack tightly on top of each other. Think also back to our Unit 1 stuff and things that you had from your Chem 100 days where we're talking about intermolecular forces. So intermolecular forces, we're talking about the idea of things like London dispersion forces if we're talking about the hydrophobic tail. The less physical contact each tail can have with the other tails in the fatty acids that are stacking together, the weaker the intermolecular forces would be for those. So think about it again. If we have kinks in our long chain tail, they can't lay as close, physically as close to each other as they could if they were all flat. And that if they are not able to be very close to each other, they're going to have weaker intermolecular forces. And if there are weaker intermolecular forces holding these molecules together, they're going to have lower melting points. This is why a significant number of fatty acids are liquids at room temperature. Melting point will decrease as the number of carbon-carbon double bonds increases. So again, the more double bonds we have, the more kinkiness there is to the tail. Therefore, the more weak the intermolecular forces are and the lower the boiling point and melting point. Therefore, more liquid at room temperature. So here's an example of that linear versus cis versus double cis, right? So, and remember that it isn't going to maybe just happen in one spot. We could have a kink here as well. And so what will end up happening is the more and more these drastic the kinks get, the less and less interactions can occur as these stack up on top of each other, right? So here's stearic acid. This is found in animal fat, which is a solid at room temperature. Oleic acid and linoleic acid. These are both going to be oils at room temperature. So when we say something is essential, we'll see this a few times in this unit. When anything is essential, it's something that our body cannot make, is not synthesized within our own body, and needs to be obtained through our diet. So linoleic acid and linolenic acid, these are omega fatty 3 and 6. Acids, again, these are things that we cannot produce on our own and that we obtain through our environment, through our diet. Um, they are used to produce quasi-hormone-like substances that help regulate things like blood pressure, clotting, immune response, inflammation. So clearly very important that we are able to obtain these fatty acids. It is possible to convert omega-3 into omega-6 and vice versa. There are enzymes in our bodies that are able to do that. So again, I always encourage you to pause the video, try and answer the question on your own, and then come back and check the answers. So we've talked about fatty acids. We're going to move now on to the actual concept of kind of like a fat and an oil. How do we get these? So what we're going to talk about next is the concept of a triglyceride. So a triglyceride or a triglycerol, depending on how you learned it, will be 1 unit of glycerol, which we'll talk about what that looks like, that will be linked via an ester bond to 3 different fatty acids. So this is what we were talking about when we were talking about stacking them. If those fatty acids were straight, like these bars are, they would lay very close to each other. However, if these fatty acids had kinks to them or severe bends to them, all of a sudden they wouldn't be able to lay as close to each other and we'd have significantly weaker intermolecular forces going on. And then if we had multiple triglycerides, they would be even bulkier, if there were points of unsaturation, they would become even bulkier where they wouldn't be able to lay on top of each other. So here's an example of how a triglyceride is formed. Here's our glycerol molecule. Glycerol is pretty straightforward. It's 3 carbons with a hydroxyl group at each carbon. That's it. And here we have 3 molecules of stearic acid. So we have our 3 fatty acids that will link to the 3 hydroxyl groups of the glycerol. 1 glycerol, 3 fatty acids. And what we have highlighted here is the esterification reaction that's going to occur, right? This is an alcohol. This is a carboxylic acid. We know that carboxylic acids can be combined with alcohols. We get a byproduct of water and an ester group. So these are ester formation reactions that are occurring to, in total, make 1 triglyceride. So when we're kind of distinguishing the different types of lipids and we're talking about the difference between a fat and an oil, typically fats are derived from an animal source, are going to be solids at room temperature, and for the most part, because they're solids at room temperature, that would mean they're pretty much always going to be made of triglycerides of long unsaturated fatty acids, so those straight flat fatty acids. Oils, on the other hand, typically will come from things like plants or some fish sources, seafood sources, liquids at room temperature, and they're composed of triglycerides that will have unsaturated fatty acids, which will allow them to have lower melting points because of weaker intermolecular interactions occurring. So here's just kind of a breakdown to visualize for you the ratios of saturated to unsaturated fats when we look at things between fatty acids of animals or animal-based products and plant-based products. So obviously, we know that excessive dietary fat can sometimes become an issue. We can develop chronic diseases from this and it can play a role in raising your cholesterol level. Obviously, if you have far too much fat in your body, you can even develop gout, which is incredibly painful. Again, I always encourage you to pause here and answer this on your own before moving on. So when we have these triglycerides, what's going to happen to them? We ingest something like this, right? So something like this triglyceride here would be ingested. How does our body utilize this? Like we said, we know that Lipids are a good source of energy and can store energy very well. So how are we going to get anything out of this? Um, Really, the first step would be breaking apart the ester bond that was formed in the making of this. So we have the three ester bonds here. They will be broken apart. In our bodies, this is done by an enzyme called lipase. In a lab, this can be done under acidic conditions. But essentially, we're going to cleave all three ester bonds, reforming our glycerol and our fatty acids. If we are to take them apart under basic conditions, remember, just like we learned in chapter 16, or what was that, 15 in unit 2, if we have esters and we break them apart using a strong base, we'll get our glycerol back out, which would be our alcohols, but we can no longer produce those acids because we're under basic conditions. So an acid can't exist under basic conditions. Instead, we're going to end up with the salt of our fatty acids, and the cation will depend on what cation was in the base. This is the basics of saponification, or the making of salts, of soaps. We have glycerol and we have salts of our fatty acids. Depending on the cation used in the base, this will either produce a hard soap, so like a bar of soap, or a soft soap, like a liquid soap, like a squeezy bottle soap. Traditionally, soap making used animal sources, so animal fat and lye, which is a crude form of NaOH. Either that or wood ash, so basically the remnants of like a fire. Um, Now, obviously, soaps are made with a lot of different things. You can find things that have shea butter, palm oil, which is not great to use. But again, these would be all oils, or any source of a fatty acid is required to make soap. It doesn't necessarily have to be animal based, but a fatty acid and a strong base. We can also go through and manipulate fatty acids with their saturations. So, unlike stearic acid, which we know is straight and has no double bonds in it, we know things like linoleic acid and linoic acid have multiple double bonds in them. And this creates those kinks. What we can do is decrease the degree of unsaturation. So we can remove some of those double bonds, which in turn would increase the melting point. Now you may be wondering what purpose would this ever have to increase the melting point of something by removing double bonds? But for most of us, we've probably at some point or another experienced eating margarine. Margarine is not real butter, but it's a desirable kind of intermediate, right? Margarine is made typically from oil. We know that oils are liquids at room temperature. Margarine, however, is not. Margarine is a soft, creamy, spreadable solid, which is the desired outcome. And the way this is done is by removing degrees of unsaturation within the oil to increase the melting point, bringing it to a semi-solid, soft, solid form. When doing this process, right, if we're breaking apart double bonds and resaturating them, we would be doing hydrogenation. Hydrogenation is the process of adding hydrogens across that double bond in the alkene. If you push too hard and you hydrogenate too much, you will end up with a very hard, waxy product. Partial hydrogenation, though, this is where we get partially hydrogenated, canola oil and stuff like that to make creamy, smooth fats, which again would be something that we desire in terms of like a margarine, like a spreadable, not really butter, but buttery type product. Some fatty acid molecules can be reconfigured to go from cis to trans. This is another way in which we would increase the melting point of the fatty acid. So a cis double bond would create a kink. A trans double bond, though, even though there's a double bond there and it's unsaturated, a trans double bond will have that tail maintain most of its linearity, which again will allow it to stack on top of itself. This is where we get the concept of like a dangerous trans fatty acid. Trans fatty acids are similar in their physical structure to that of saturated fats. And therefore can go through and stack more readily and be solids at room temperature and follow that same path. So here's an example of that hydrogenation where we're partially hydrogenating one of the fatty acids in this triglyceride. Don't worry too much about the nickel here. Hydrogenation typically needs some sort of metal catalyst for it to work. It's kind of a site at which the alkene sits upon and the hydrogens kind of can interact with them there. Sometimes it's with nickel, sometimes it's with palladium. Don't worry about too much different metal catalysts. Just understand that hydrogenation is a way that we could make a more saturated triglyceride. If we had a base, this would be making soaps where we would break those ester bonds and we would end up with the glycerol and the salt of the fatty acids. If we have acidic conditions for the triglyceride, we break apart the glycerol and we would get our acids out because we were in acidic conditions. As always, I encourage you to pause here and give this one a try. So we've talked about the concept that if we hydrogenated a glyceride too hard, a triglyceride too hard, that we would end up with a wax. So we're actually going to talk about what waxes are. Waxes are just going to be esters with really long fatty acid chains on either side of them. So again, here's our ester, the C double bond O carbonyl and the sandwiched oxygen. And then on each side of it, we have very, very long chains of carbons now instead of smaller ones that we've seen. These waxes are going to be seen on everything from skin to fruit to leaves to fur, basically any natural wax that is derived from a plant or an animal will exist as these long chain esters. It's also very important for things like making candles, cosmetics, protective coats on furniture, cars, all that kind of stuff. We also talked about the idea that lipids are essential for structure. A lot of cells utilize lipids as a way to kind of develop a membrane that kind of either separates the whole cell from everything outside of it, keeps all the stuff inside the cell, and the stuff outside the cell outside, or it can be even used to partition up portions of the cell. So in prokaryotic cells, right, these are going to be the little one-celled organism type things. They don't have any specialized bits and pieces to them inside them. So they are very, very simple and just have an outer cell membrane. Eukaryotic cells, however, kind of separate out the different portions of the cell and utilize membranes not only as a cell wall on the outside, but also within it. And then in addition to that, sometimes organelles or like little structural features are also a part of lipid formation. So again, this is just kind of, this is, I understand, a little more bio-heavy than you probably thought you were going to get in this class, but we're just going through some of the concepts of what organelles there are that are made up of lipids or have lipids in them and what their function is. So most membranes are going to be predominantly lipid and peppered in with proteins. And those proteins have jobs to do. Some of them are like salt channels that will be kind of the egress ingress into the cell and out of the cell. Um, we have three basic types of lipids that make up these cell walls: cholesterols, sphingomyelins, and phosphoglycerides. And we'll talk about each of those in a little bit. The concept, which is really important for humans and basically all mammalian life, is that we have a fluid mosaic model. Our membranes are modeled in a way that they're flexible. This lipid bilayer has some give and take to it. So, for example, think if you took a stalk of celery and you thwacked it relatively hard on the edge of a table, that celery will break. And that is because the cell walls of the celery are rigid. There's no lipid bilayer there to kind of cushion the blow. Now, I don't encourage anyone to do this, but if you were to take your arm and also hit it against the edge of the table at the same force that you did that broke the celery, in all likelihood, you will not break. And that is because your cells are able to kind of flex and absorb these kinds of impacts. This is by, again, having those hydrophobic tails extended inside the lipid membrane bilayer and the heads on the outside towards the solution, towards body fluids. Um, when the lipid bilayer is disrupted and broken apart, it instantly heals itself because of that repulsion of those hydrophobic tails on the inside. Once they're exposed to the outside, they immediately will seal up to, again, reclude water. So again, here's that concept of that lipid bilayer meaning two of them. Here we have our hydrophilic heads and our hydrophobic tails. So the bulk of the environment of a cell membrane looks something like this. We have proteins, like I said, these proteins sometimes are channels that come in and out. Cholesterol is also involved. It's kind of like a structural thing. Sometimes there's carbohydrates involved as well. But right now we're focusing on the lipids. So here again is a kind of overall visualization of what's going on with this bilayer between the hydrophobic and the hydrophilic portions of in the cell, the environment. Now remember that if this is a multicellular environment or something like that, the same concept applies, but the hydrophilic, the ones that like water, the polar heads are exposed on the outside, both outside of the cell and the pocket inside the cell. Because, right, there's still fluid inside the cell that's going to be predominantly polar. So we can't just have one individual layer where we've exposed our hydrophobic tails to that inside layer as well. The flexibility of these lipid bilayers also extends to the ability for them to move laterally against each other. So they kind of shift, just like you would slide your hands on top of each other. The membranes can do that too. They kind of float along that bilayer. Another important biological classification for some lipids would be steroids. Most steroids will follow this general, what we call motif or backbone. So three like cyclic hexagons and one pentagon. Typically, they're going to be nonpolar. They are predominantly hydrocarbon based. Now, they're decorated as well, but they're going to be nonpolar, and they include steroids such as cholesterol, bile salts, things like that. Cholesterol is the most abundant, even in a healthy individual, steroid in the body. It is a part of cell membranes, as we saw in that previous picture. It is a precursor to other steroids, so it's kind of a building block for other steroids. It's also present in food we ingest, but we also produce it ourselves. So cholesterol is not essential. We are able to produce it in our livers ourselves, but we also ingest it from what we eat, particularly animal-based foods. Um, Another steroid, again, so you can see, right, we have significant amounts of decoration going on here, would be bile. Bile is the yellowish, kind of greenish secretion that's stored in the gallbladder. So if you have gallstones and have a gallbladder removed, you are going to have greater difficulty digesting different types of food, particularly very fatty foods. So when a healthy individual who has their gallbladder and properly functioning bile eats fatty foods, they enter the small intestine, and the gallbladder will empty bile into the intestine to help break it down. Someone who has their gallbladder removed obviously no longer has these stores of bile, and therefore don't digest fatty foods as well. So bile acid salts, like we said, they're a salt of the steroid, and they help break down these lipids, right? When we're talking about fatty foods, they break down lipids into smaller and smaller portions. And the smaller and smaller the portion, the bigger and bigger the surface area there is in comparison to the size of the molecule, which allows hydrolysis to occur. So someone who is experiencing gallstones, there are two different kinds of gallstones. There are cholesterol-based gallstones and there are mineral-based gallstones. Typically, women tend to get more of the cholesterol-based gallstones, where men get more of the mineral-based, but that's not a hard stop one way or the other. There are plenty of women who have mineral gallstones and vice versa for men who have cholesterol-based ones. Either way, gallstones can be excruciatingly painful. They can also become incredibly dangerous if one is to become lodged or kind of stuck in the bile duct. Um, you can have pain, nausea, fever. You can actually start kind of losing color to your skin. Your poop will actually become gray. It's very strange because there's no more bile in there pigmenting it. So right, our stool is simply all the waste left at the end after we've drawn everything out of it. So you would end up with gray poop. Typically, when you have a gallstone, they just remove the entire gallbladder and not necessarily go in and take out just the stones. It would depend on the individual and the circumstances surrounding the gallstones. So we also have steroid hormones. These are probably more what you're familiar with when you think of steroids. They're secreted, they're messengers, right? So they tell our bodies to do things and trigger certain responses. So we have things like the stress hormone cortisol. So this is a part of our fight or flight response. Estradiol, sex hormones, adrenal hormones. Basically, these help regulate everything from our body fluids to how much our electrolytes in our body are regulated through this, carbohydrate metabolism, all these kinds of things. So it's really, really a important aspect of our physiological health that we have steroid hormones. Again, here's cortisol. These are adrenocorticoid hormones, sex hormones, so produced in either the testes or the ovaries. And these will, during puberty, help develop secondary sex characteristics. So here we have testosterone. Again, you can kind of base off of the name. We see own, we're mentioning that because of the ketone on here as well. Anabolic steroids, these are going to be things that are going to be uh, often misused or abused, and they can promote growth, obviously, is what they're often used for, but the side effects can range from acne to testicular atrophy, lowered sperm count, temporary infertility, or permanent, depending on how much usage there is. Um, so these are often utilized as a way to kind of hack a body into growing more muscle. Female sex hormones, clearly important for the reproductive process, but things like estrogen, progesterone, estradiol. These hormones will help a woman go through her fertility cycle. Also, certain of these hormones will uptake and either downtake depending on pregnancy or not being pregnant. So again, just when we look at these, we can see the general structure of this steroid in each of these. Now, it changes, right? This is an aromatic group here, where this is not aromatic. This one is going to be a diol, right? There's two alcohols on this one, where we have a ketone on this, two ketones on this one, but ketones on these two. So the general backbone of the steroid remains the same, but the decorations around it vary greatly.